Catalysis Biosynthesis chapter 3 from our book. And so we had just kind of started off a little bit with just reviewing laws of thermodynamics, talking about how pathways are linked and how our metabolism is the sum of the catabolic and the anabolic pathways together. And so last week we had ended at that point with starting to look at photosynthesis because it is that coupled system where you're looking at both the catabolic and anabolic mechanisms all working together to provide the energy to that plant. So we're going to go ahead and get started from that point. So obviously our photosynthetic organisms are really cool in this way because they can use the sunlight and these inorganic substances and the organic substances to synthesize their own organic molecules. They can create the sugars that they need, and then they can break them down in the second stage to get the energy out of it as well. So here you have this 2 stages of that first part, the photosynthesis of preparing the sugar, where they capture light energy from sunlight. They also utilize water and get, and release oxygen in that first stage. They utilize all that energy that they have stored through these processes through activated carriers in the form of ATP and NADPH, which are then transferred into the second stage, which is their main process of making sugars. They're going to utilize all this energy, the water and the carbon dioxide coming into the system and prepare sugars out of them that they can then utilize for energy consumption down the road. So this is actually, one of the main ways that we have carbon atom cycling all throughout our biosphere continuously. So again, going with the laws of thermodynamics, the total energy, the total mass, the total matter, all those things are remaining constant. They're just moving from one place to another. They're moving from one system to another as that energy is needed to be utilized by one thing or another. So here you can see an example of how this food chain works, where you're taking in, the carbon dioxide in the atmosphere, the water in the atmosphere, you're using the organic matter dissolved in the humus, and you're taking all this information, all this matter, and you're making food out of it through photosynthesis, and then utilizing it through breakdown of that food. And it could be by other, by plants themselves that made it, but it could also be by other consumers, by the algae, the bacteria, the animals that are going to consume those plants as well. So cells essentially are obtaining energy by the oxidation of these organic molecules. We are going to be talking about that process more and more in detail in this chapter. So you have your photosynthesis component, which is the preparation of these sugars from organic sources that are from the organic molecules and from the matter that they are containing. So you have the typical equation that we always look at, the carbon dioxide plus water gets converted into oxygen and sugars with the utilization of the sunlight for energy consumption, and that's going to be all your photosynthetic organisms in the form of plants, algae, and some bacteria. These sugars and the other organic molecules that are prepared then go into cell respiration, where they are broken down for their components. And that energy that is released from those breakdown of products is then taken into the form of ATP and NADPH, which is the useful chemical bond energy that can be utilized for those organisms to do the work they need to do. So they're, just reciprocal of each other at the end of the day. Here you're taking in the water and the oxygen, and in the cell respiration component, you're going to be releasing those same things as you break down your systems. So all this stuff essentially involves a lot of oxidation and reduction reactions, basically transfer of energy in the form of electrons from one place to another. At the basics of that, should be reviewed, obviously, from chemistry. Anytime you have loss of electrons from one molecule to the other, it's going to be called an oxidation. And anytime you have gain of electrons, it's going to be a reduction. So here you have an example of these two atoms, both with one valence electrons. When they come together to form a polar covalent bond, the one atom that was smaller and ends up by having a partial positive charge because the electrons are spending more time with the other molecule or other atom will be what you would call an oxidized part of the molecule, and the partial negative charge component would be the reduced part. So whatever has the negative charge is going to be the reduced one, and the positive charge is going to be the oxidized one. Here is another example of how reduction happens. And reduction occurs more and more as numbers of your carbon-hydrogen bonds increase. So here you have an example of how you have your carbon dioxide molecule all the way up to methane. The more complex, the more hydrogen bonds are there, the more reduced that molecule or atom is going to be. And the less hydrogen bonds are there, the more oxidized it typically is. So oxygen obviously is a larger, molecule, a larger atom. It's going to have, you usually end up with that partial negative charge versus hydrogen. It's small. It usually has that +1. So you can use that to kind of remember that as well. So the use of energy by cells basically is going to be by cells are not going to make just some de novo energy from nowhere. It has to come from whatever is already present, the matter and the atoms that are already there, the energy that's already there. All they do is they convert energy from one form to another so they can utilize it for their own work. Photosynthetic organisms can obviously utilize the energy from the sunlight to make their own organic molecules that they then break down to make energy for their other function, for their metabolic work. And all of that work usually involves oxidation and reductions. That is just electron transfers from one molecule to another or from one atom to another. So at the end of the day, these kind of reactions can happen all the time in a chemistry lab, in another lab, in the environment, but they usually require some kind of energy input. There is some kind of what we call activation energy that is needed to get this whole process started. This activation energy can depends on what type of reaction it is. Some reactions will require a lot lower activation energy than others, but every reaction will require some activation energy before that process will happen. Well, that activation energy is sometimes not able to be provided at physiological temperature many times in our body at our physiological temperature in our metabolic processes. And so our systems, our biological living systems require the reduction of that activation energy requirement in order to be able to do the work they need to do. And that is done through the help of enzymes. So even when the reaction should happen, it's a very energetically favorable reaction. It's still going to be requiring some activation energy to get started. To, even when you call those so-called spontaneous reactions, they still need that. So to get to that point, we utilize enzymes, and these enzymes can then lower that activation energy so that we can run those reactions at physiological pH, physiological temperature, and move them forward and get the work done that we need to do. So in general, when we are going to do this process, you are going to have your reactants at a higher energy starting point. They are going to go over this hump of activation energy, depending on whether the enzymes are there to lower that activation energy or not. And the new products are going to be at a lot less, a lower energy component, and that's why they happen to have, occur. So most of chemical reactions are going to proceed in loss of energy at the end of the day, because that useful energy that's released from the process is what you need to do work at the end of the day. Okay, so this type of stuff essentially, again, has to still follow the same laws of thermodynamics, and enzymes do that. Enzymes obey those second law of thermodynamics. They increase the rate of your energetically favorable reactions, but they do not they are not going to be adding in any other component to that. So reactions that are going to produce disorder in the universe are generally going to release lots and lots of free energy, and they tend to be spontaneous reactions. So they're going to not require as much help from enzymes as those that require an input of free energy. So those that are non-spontaneous reactions that need more work because they are going to give you products that are at a higher energy level than beginning are going to not be energetically favorable on their own, and the enzymes cannot force that to happen. They can only increase the rate of reactions that will happen spontaneously anyway because they're energetically favorable. So just because you have enzymes doesn't mean they can just make any reaction happen. It still has to be an energetically favorable reaction. So it should still release energy at the end of the day, and it should still give you a spontaneous reaction at the end of the day. So sometimes you will notice that, when we start talking about reactions, that's not the case right off the bat. There are portions of your metabolic pathways that are energetically unfavorable. But by the end of the day, when you get to the sum of all the reactions in that pathway, you're still getting to an energetically favorable component. So lowering the activation energy obviously greatly increases the probability that a system can actually do that process and make that reaction happen. Right? Because otherwise, if you cannot lower it, then it's going to be a lot harder for that system to do that work and to make that process happen. And that's all that the enzymes are going to do. They're going to provide just the right environment so that you can lower the activation energy barrier. Sometimes they do that by providing a catalytic site where the substrates can bind and be in close proximity so that the reaction can happen more easily, or they provide the right pH or the environment that is needed for that stuff to work. So in an uncatalyzed reaction, essentially, this is an example of what that might look like. The waves are just not large enough to get, right? You just don't have enough energy in the system to get over that hump to get enough activation energy. But in a catalyzed reaction, you're going to help the system pass over that hump so that you can get the reaction moving forward. Another thing with enzymes is that at the end of the day, they remain unchanged. They are not utilized themselves in the process. They do the work and then they're free to go do it again. And they can keep doing that process as long as they're around. So in enzymes linked reactions, the enzyme will bind to a substrate, whatever it is that it's going to be affecting. It's going to make an enzyme substrate complex that is going to then follow and go through the catalysis, do the reaction that needs to happen. At the end of the day, you will have an enzyme product complex once the reaction is complete. This usually has a much slower or much lower binding efficiency to the enzyme than the original substrate did, which causes it to be released. And now the active site is open again, ready to bind to a new substrate and go through the process all over again. So what exactly what exactly determines whether or not a reaction can occur is something called free energy. The free energy is basically a usable energy that are present in the system. And the free energy of your energetically favorable reaction has to have higher free energy in your substrate than in the product at the end of the day. And the free energy change, which would be the change in this energy once the product has been made, can be negative or positive. But in order for a reaction to be spontaneously occurring, it has to be negative, right? If it's not negative, then the disorder of the universe will increase, right? And it won't work the same way. So for energetically favorable reactions, you want your delta G, or the change in free energy, to be below zero or negative. So negative free energy production is going to give you energetically favorable and spontaneous reactions. If you have a positive energetically positive delta G, then it's going to be energetically unfavorable, because obviously that's going to require more energy input, and that won't be a useful product in that case. So these reactions can occur only if they are coupled with another reaction that brings this overall delta G down to a negative. And they are not going to happen all by themselves in isolation. And that's usually what happens in our body systems, that we link those energetically unfavorable parts of our metabolic processes to a really highly energetically favorable part of the metabolic process, so that the overall sum of those two processes are still negative delta G. Any questions so far, guys? Not even if they are enzyme activated. Is this now? Yes, not even if they are enzyme activated. They have to have a negative delta G. Is that what you were talking about, Janelle? Yes, even if they're enzyme, enzymes cannot activate a non-spontaneous reaction just out of nowhere. It has to be linked to it has to be coupled with a spontaneous reaction for it to even go forward. Enzymes still have to follow the second law of thermodynamics, so they can't just make that happen out of nowhere. And we'll see an example in a little bit. So here is an example actually right here of how reaction coupling can help exactly what you are just talking about. So when you have reaction coupling, you basically utilize the energy from a really high spontaneously occurring, right, really negative delta G producing reaction to drive that second reaction that is unfavorable otherwise, that is non-spontaneous otherwise. It's not going to happen on its own. No enzyme can make it happen all by itself. So the energetically favorable reaction is what's going to happen usually first. And that's going to produce a lot of energy that will then drive that second reaction. That's going to be the positive delta G1, right? And that's going to utilize some of that energy, but still have some left behind. And at minimum, it's going to zero it out, if not leave some behind. And so the net free energy change for the pair is usually less than zero or at most zero. So because of that, no matter what reaction you're looking at, it could be an energetically favorable one, it could be a coupled one, it could be a non-spontaneous or spontaneous one. Every reaction is going to have in some kind of an equilibrium state. For energetically favorable reactions, that equilibrium state is going to be very, very skewed towards the product side, because that's where the energetically favorable reaction is happening, right? That's where the lowest energy is. And on the other hand, if you are looking at a non-spontaneous reaction, it's going to be heavily set towards the substrate side. So it's going to be a very, very slow moving reaction on its own, if at all. However, when you get into coupled reactions, that's how you move that process forward and make that reaction have a little bit higher equilibrium state towards the product. That's how you push it towards the product side, by coupling them. So for each individual molecule, you would have to think about how that ratio changes, right? So here you have, an energetically favorable reaction where you have your substrate Y and it's getting converted into X. So majority of that reaction, because it's energetically favorable, it's producing negative delta G. It's going to be towards the X side, and very little, if any, of this product is going to go back towards the substrate side, can go back to the other one, right? However, you can get to a point where there's large enough product available that at this point there's just not enough substrate left, essentially, right? There's just nothing left, or very little substrate is left, and you'll see a slowdown of the reaction. So that's going to be that equilibrium state where there's going to be very little substrate left, and most of it is already converted into product. So there's no net change at that point in the net ratio of Y to X. They will be stable at that point. Until they reach that equilibrium state, you will still see increase in X happening over time in a constant manner. It just slows down as you get less and less substrate available for that product to be converted into. Yes, we're going to get to that as well later on, that the ATP, GTP is going to come in here as well. okay? So here is what we can do. We can calculate the standard free energy change of a reaction by looking at the equilibrium constants. And so, so there's a Gibbs free energy, and what we can do is basically we can look at the reaction coefficients between our substrates and our products, and we can calculate what the total equilibrium of that reaction is going to be. The basic equation for that is essentially the change between reactants and products. And so as long as we know those numbers, we can calculate the equilibrium constant for that particular reaction. Why is this? Sorry, my computer has this like weird. I'm trying to get this control bar out of my way, but it's not working. Okay, so changes in the free energy of sequential reactions when they are coupled are going to be additive, right? So if you just have a single reaction, that's fine. You can quickly calculate its Gibbs free constant and you're done. But if you have coupled reaction, then you have to add the individual reactions together, and they're going to all add up, whether they're positives or negatives. And your final answer is going to be the sum of all those different reactions. And so for each one of those, you would need to know the equilibrium point for that reaction. So in this case, there are two different reactions. They're linked together. The first one, let's look at that. So you have your X and your Y. So X in this case is my substrate and Y is my product. If that is the case, then which way is this reaction? Is this an energetically favorable reaction, or is it an energetically unfavorable reaction based on the arrows, looking at the thickness of the arrows? Why is that? Yes, it is unfavorable. Why is that? Why would you say that it is an unfavorable reaction? Yes, exactly, because it takes more energy, essentially. It's a delta G positive reaction. So this one is not an energetically favorable reaction. It wouldn't happen unless it was linked to something very, very highly energetically favorable reaction. And that's in the second part. You have the Y to Z reaction, where even a tiny amount of substrate will give you a lot of this product, right? And that's where this is a very energetically favorable reaction. So anytime you're getting this product, it's immediately getting sucked up and getting used to make this. That's going to force this reaction to move forward and keep producing more, because it is going to produce this high energy product. So that's why, coupling helps. But when that coupling is happening, you will need to know the Gibbs free energy for the first reaction and the second reaction, then add them together to get to your final answer. So here is an example of something like this. So this is the, free energy change. These are coupled reactions. And in this case, you have the single reaction where you have glucose and fructose coming together to produce a sucrose molecule. And this one has a Gibbs free energy change of 23 kilojoules per mole. So this one is not going to happen all by itself, right? Because obviously this one has a positive energy change. It's not going to be energetically favorable. However, if we couple it with something really energy producing, in this case, coupling it with ATP hydrolysis, essentially breakdown of ATP into ADP, the reaction becomes suddenly favorable. So we can utilize the energy from ATP by breaking it down and releasing that phosphate. That's going to give us a negative 30.5 kilojoules per mole delta G. And that's going to offset that 23 and give you a final reaction when you couple this. So you have your glucose, you add the ATP energy, the phosphate comes off, adds to the glucose, changes it to glucose 1-phosphate plus the ADP, and then that becomes a favorable reaction. So you get your sucrose and the phosphate is back out. The total net change of energy is going to be the addition of those two numbers, and it will be the negative 7.5 kilojoules per mole. So your net free energy would be the sum of both of those reactions, whether they're 2, 3, 5, or 10. So glucose metabolism, for example, has 10 linked reactions, and all of those would be combined together to get you that final answer. Okay, so the sucrose is made in a reaction completely driven by that hydrolysis of ATP. If that wasn't there, it wasn't going to happen. You were not going to get a sucrose molecule out of it. Did that make sense, Rhoda? I think that's what you were talking about earlier. Yeah. Questions about this? Okay, cool. So the equilibrium constant is basically what we are going to use to measure the directionality of a reaction. The equilibrium constant is going to, again, be dictated by the delta G changes as a reaction goes from A to B, your substrate to product, and that equilibrium. The point where it is the lowest, that equilibrium, is at present, is going to be your equilibrium constant. So here is an example of pure A substrate or pure B product. The equilibrium constant, if the reaction was equal on both sides, would be 1. That would be that same amount of product and substrate are present at equilibrium. When things shift over one side or the other, that equilibrium constant obviously is going to shift along with it. So it will be higher for when it's shifted towards the product side, and lower if it's shifted towards the substrate side. This equilibrium constant is going to basically indicate to you how strong those molecular interactions are going to be in your substrate versus the product, right? So that's your association kind of rate, right? How well are they how likely are they to associate with each other to produce that product that you want? So you have your association rate is basically going to be your the constant for that particular, the equilibrium constant for that particular reaction, multiplied by the concentration of your different substrates that are present. So in this case, concentration of A and concentration of B. Combining them together, you would get your, at equilibrium, the rate of reaction. So your association rate is going to be dependent on the concentration of each one of the substrates that make the product in that case. So the equation that you would want to look at for equilibrium would be the concentration of your product divided by the concentration of your individual substrates. And that's going to essentially be your equilibrium constant for the reaction to move forward in the product reaction divided by the equilibrium constant for the reaction to be switched off completely. And that's your final equilibrium constant. So very small changes in the number of weak bonds can have a very, very big impact in how well the binding efficiency works. And here is an example of that. So, in both cases, the molecule numbers are very, you have the same number of essentially molecule A and B, and you're getting your complexes. But look at how different those complexes are if you have different equilibrium constants, right? If the association constant, how well those binding works that are different. So in the first case, the concentration of both is 10 to the negative 9 moles. And to calculate the equilibrium constant, again, you're going to take into account the concentration of each one of these molecules. And you get a lot more product in this case, compared to the second one where the equilibrium constant was weaker because they had less binding efficiency. You will be provided with enough information to, yes, you should be able to calculate, given something like this, given the molecule concentration and the equilibrium constant, you should be able to figure out which one is weaker, which one is stronger, and also the equilibrium constant. Yes, using this equation. So in the second instance, just because the equilibrium constant is weaker, even having so many more, 4 times more molecules of A and B does not give you a lot of complex because the association is very weak and they are just not able to bind and stay bound. So in that case, this is going to be an example of an energetically unfavorable reactions, while the first one is going to be an example of an energetically favorable reaction. So again, given something like this, you should be able to tell if it's an energetically favorable or unfavorable reaction. Now, what else can dictate how enzymes help the reactions, or what can drive the energy forward and make that reaction happen, are some other things outside of enzymes as well. One of them is thermal motion, how well they're moving in that space, how much they can travel, what the velocity is in that area, what the heat is in that area. So this kind of thermal motion allows enzyme to find their substrates to bind to them. Now, you have to be careful with that too. Sometimes when there is too much heat in the system, it's not going to allow for that binding to form properly because the molecules are too fast in motion. Similarly, if you're too slowed down because of cold temperature, because the temperature has gone down too much, it's not going to be able to find, they'll be frozen in there, or they will be not able to find each other for enough time to bind and stay working. So enzymes need typically optimum temperatures, optimum pH to work effectively. Here again is just a reminder of how enzymes cannot change the equilibrium point for reactions, but can just make it happen quicker. That's all. So even when you have an enzyme catalyzed reaction at equilibrium, it's going to look the same as an uncatalyzed reaction at equilibrium. It just would have gotten there quicker. That's it. It just makes it go faster because it lowers the activation energy and makes things happen a lot more efficiently. That's all. So it's just changing the rate of reaction, but it's not changing the final outcome. It remains the same. So if there were going to be 100 molecules of product to 2 molecules of substrate, that's going to be always the case. So what are some things that are going to dictate how well the enzyme works? Well, one of them is going to be dependent on its ability to process the substrate. How quickly does it work, right? So the rate of reaction is going to be dictated by how quickly it can bind and process the substrate and make it into product. And so each enzyme will typically have, again, a linear region where it's the more enzyme you add, the more quickly it's going to work. And then eventually you're going to start reaching that saturation point, and eventually it's going to reach its maximal capability. That maximal capability is called Vmax. And typically the rate of reaction is, the fastest at half Vmax. That's the point where it's reaching at the fastest rate, how rapidly it is processing the product. And that's also related to the kinetic constant. So your substrate concentration, as well as the amount of your enzyme, are going to dictate where that half Vmax is and where the Vmax is. Typically, the more substrate you add, obviously there's more substrate available, so the enzyme can keep on working and keep on churning out more product, but it's still going to be, it's going to be dictated by how rapidly it can process it. If it can only process 1 molecule per 5 minutes, it can only process 1 molecule per 5 minutes. You can keep adding more substrate. It's not going to make that rate go any faster. It can't go above the Vmax. That's the limit of where it goes. So most enzymes display what we call Michaelis-Menten kinetics. When there is very low substrate concentration, it's going to be roughly proportional to substrate concentration. So the substrate concentration will dictate how fast the reaction goes. That's the linear portion of that graph. That's that, where we were still in that linear region below the Vmax, half Vmax. When you have really high substrate concentration, it switches. It switches to how much enzyme is present. It's going to be more dependent on the velocity of the enzyme. How fast can it work? And that's going to be dependent on not the substrate concentration, but just the constant Vmax, the maximal ability of that enzyme to work. How fast can it do it? So your Michaelis constant, or Km, is going to be the substrate concentration at which the reaction proceeds at 1/2 maximum velocity. That's the highest essentially a most efficient point in your reaction is what we call the Michaelis constant. They're going to take into account substrate concentration at the time when there is half maximum velocity of the reaction going on. You should be able to calculate that given a graph like this or given the information about the reaction. So here is an example of how you would see that happening, right? So when you add increasing substrate, you will get more and more and more product, obviously, but there's a limit. Eventually it saturates out. They're just so fast that the enzyme can work. So here you can see the velocity can be calculated using that as well. If you have the Vmax and the substrate concentration, and you are given the Michaelis constant, you can calculate the maximum rate of reaction for that substrate consumption, how fast that enzyme can work. Similarly, you can also use the same equation to calculate 1/2 maximum velocity by just rearranging the equation to match for that. Seriously, I don't know why it does. Questions about this? Okay, so obviously knowing this, we can control how our enzymes work, right? In a test tube especially, we can look at enzyme kinetics, we can calculate all these constants anytime we are looking at a new inhibitor or new enzyme and a new reaction. We can also learn to control it. We can learn to enhance or inhibit their function using specific compounds or products. So enzymes can have different types of you can inactivate them by using competitive or non-competitive inhibitors that bind either directly to the active site or away from the active site and stop the binding of substrate to your enzyme so that no product is made, right? So here is an example where you have substrate only and the reaction moves on at a very fast rate at the normal Vmax. You can tell that in the red line, it is not reaching the Vmax because you have the inhibitor present. And the inhibitor is going to bind to some of the substrate so that no more, or not substrate, bind to your enzyme so that the substrate can no longer bind to it and lower your high efficiency of that enzyme. Similarly, down here, you can see how that would affect the 1/2 Vmax as well, because now that there is inhibitor present, it's going to require a lot more time to get the same work done, essentially. So just to review this portion, chemical reactions proceed in the direction of your loss of free energy, right? So you want to see a negative delta G. Enzymes that reduce the energy still need to, they're still going to be working with spontaneous reactions. And if not, they need to be coupled with a spontaneous reaction in order for that reaction to happen. The free energy change for a reaction determines when it can happen or if that can happen. The delta G changes as we move toward the equilibrium, obviously, because as the reaction gets closer to equilibrium, it's going to slow down. So it's not going to be as energetically favorable anymore, eventually reaching an equilibrium state. If the reaction is equal, both at equilibrium, if the reaction has the same amount of substrate and product, you're going to see a K of 1. Otherwise, it's going to be shifted one way or another. The standard free energy change is what's going to be dictating whether the reactions are going to happen or not. In that case, you're adding up the sum of all the reactions in the process to get that total change. Also, so that's actually that, in complex reactions, the equilibrium constant will also need to include your concentrations of reactants and products, and also the strength of the bonds that are happening in the product once it's getting made. And the stronger the binding association, the better, easier it's going to be for the more energetically favorable that reaction is going to be. Even minor changes in that association binding will lead to greatly reduced product production. Okay, questions so far? Yeah, recording. Yes. Okay. So not every reaction is going to happen just simply with the presence of enzymes. Some require even more help. And these come in the form of activated carriers that are required for biosynthesis. An activated carrier is essentially anything that can provide energy to the system to allow that reaction to happen. And so that's when you have these, again, coupling of your catabolic and anabolic reactions. Your anabolic reactions, if you remember, are the ones that are energetically unfavorable. They need energy input in order to move forward. And your catabolic reactions are going to be the energetically favorable ones. So in activated carriers, you basically utilize these activated carriers like ATP, NADPH, GTP, all those things to run, they take those catabolic reaction products and utilize that energy to then run the anabolic reactions and make them move forward. The biggest activated carrier, of course, is ATP. That's the most widely used activated carrier. It is your adenine ribose chain with 3 phosphate bonds. This last one is the highest energy bond that produces a whole lot of energy. And that's going to be what's going to give you that energy to run those reactions. So this energy can be taken in from sunlight or from breakdown from food. Either way, that's what's going to be put into that bond as it is put together. And then when you break it up, that's going to be what's going to be releasing that energy. That's going to be your release of energy, right? That is available to drive those unfavorable reactions. And this inorganic phosphate is then released as the byproduct. The ATP hydrolysis is usually tied to phosphorylation of another molecule. It just doesn't happen out of nowhere. So it's not like you just take that phosphate group off and then go on with life. Typically, you use that phosphate for something else. You're transferring it from one place to another in order to make that a relevant reaction. So here you have your, typical ATP molecule. That phosphate is going to get not just broken down, but you'll release the ADP in the molecule, but that phosphate is going to be moving to another molecule in the form of phosphoester bond, creating a phosphorylated activated molecule of some type at the other end, as you saw previously in the glucose example as well. So here is an example for one of them that, again, energetically unfavorable reactions are going to be driven by this type of ATP hydrolysis. So you have in step 1, the activation of the steps by taking your ATP, breaking it down and taking that phosphate group onto your substrate to make it into an activated molecule. That's an intermediate high energy molecule that will then, in the second step, be broken down through condensation to create your product. So in that second step, the second substrate will come in and swap off with that phosphate using that energy to create that new bond, leading to your net result of that product that you need to get at the end of the day. So the net result is going to be your, the initial substrate, the 2 substrates plus the ATP that are going to give rise to your product, plus the ADP and a phosphate group. Here is an example of that. Here you have the glutamic acid. In the first activation step, it's taking the ATP, it's creating this high energy intermediate. And then in the second condensation step, the ammonia is getting added to it to make a glutamine molecule. So that's how glutamine is getting made from glutamic acid. I don't know why that weird toolbar keeps coming today. Other, in addition to ATP, other activated energy molecules can also play a part. 2 other main ones are NADH and NADPH. They are less energy producers, but they still can be activated carriers of electrons. NADPH is the reduced electron carrier, and then NADP plus is the oxidized electron carrier. So depending on the reactions that are happening, you can use one or the other. NADH is used in catabolic reactions specifically, but NADPH, on the other hand, is more for biosynthesis of molecules at the end of the day. So just to know other forms of energy carriers as well, other than your typical ATP. NADPH and NADP plus can go between each other, kind of like ATP and ADP. So NADPH is activated carrier of electrons. It's going to go through an oil rig reaction to give you, in the process of transferring those electrons from one place to another. So this will lead to oxidation of the first substrate and then reduction of a second molecule on the other end as a result. This is very important in the final stages of creating cholesterol, where this process helps to create that final structure in the cholesterol molecule to create the perfect ring structure in there. There are other, obviously, minor molecules that are also part of activated carrier molecules in metabolism. Again, ATP is your phosphates, NADP, NADH, NADPH, and FADH2 are going to transfer electrons and hydrogens. Then there are others like acetyl-CoA that will transfer an acetyl group, carboxylated biotin that will transfer a carboxyl group, S-adenosylmethionine that will transfer a methyl group. And then obviously many times you have sugar molecules getting attached to your proteins, to your lipids, and that's done by uridine diphosphate glucose that gets transferred as a glucose molecule on top of whatever it is that it's working with. All these are high energy molecules that can be broken down to create energy that can be utilized as energy for the metabolic processes that are linked with them. Questions about that before we move forward? I was just checking how far we were in the chapter to make sure we were on track. You need to be able to identify ATP, NADP, NADPH, and acetyl-CoA. Those three are usually the main ones that I focus with. The rest of them, it's, again, open book, open notes. If you are asked a question about it, you can, but those three are the main ones that I'm going to be focusing on as far as the energy transfer and in what way they produce that. Okay. So yeah, so acetyl-CoA, it's a very important activated carrier. It is extremely important in certain metabolic processes. In this molecule, again, it's an adenine and ribose, but it's connected. In this case, your phosphate groups are not the same way. You still have 3 phosphate groups here, but there are only 2 phosphates attached to this really high energy bond at the end with the acetyl group. This group is the one that is important in its reactions. And that's the important part in there, that it's that end acetyl group that is the very high energy bonds in there. All our macromolecules, no matter which one it is, whether it's polysaccharides or amino acids, proteins, nucleic acids, all of them require a lot of energy input. All of them require one of these molecules to be working with them in order to get that energy. Most of the time, it is coming from ATP, right? So in the case of nucleosides, nucleic acids, polysaccharides, and protein, because that is the highest energy production molecule, it's coming from some kind of nucleoside triphosphate hydrolysis, whether it is in the form of GTP or ATP. Those would be the 2 main ones. So here is, again, an example of that. You have the ATP hydrolysis that can be used for pyrophosphate formation. So in pyrophosphate, you're not just using one ATP, you're using, one phosphate from the ATP, you're using 2 of them. And you remember that from when we made the DNA, right? When DNA, each time you're adding the base, each time you're adding the new part, you're actually using 2 ATPs, not a single, or 2 phosphates from the ATP, not a single one. So in those cases, the adenine triphosphate, it's going to be breaking down a pyrophosphate, 2 phosphate groups together to get that energy level that is needed to add another base into your system. So nucleic acid synthesis is going to be a multi-step process because it's going to require so much more energy consumption. And so here is an example of how you have your nucleoside monophosphate, right? And the sugar base pair is going to come with the triphosphate attached to it already. You are going to break down a pyrophosphate group, leading to the new addition of the base. And then you are going to end up with a chain which contains your next nucleoside and is ready to go on with the next. So this is a high energy process. It's not simple like your glucose energy. Okay, so just going through that a little bit more in detail. So the formation of these activated carriers is coupled to energetically favorable reactions, obviously, otherwise it's not going to work, right? That's where they get the energy, is from those energetically favorable reactions, which produce the delta G, the negative, that is going to help to put these phosphate groups. ATP is the most widely used activated carrier. The other ones that you need to know about are NADH, NADPH, that are both activated carriers of electron specifically. And then your acetyl, any other carriers, be aware of them. But again, we are going to focus mostly on the acetyl-CoA, NADH, NADPH, and the ATP, GTP combination. And obviously our biological polymers, any one of them, whether it's polysaccharide nucleotides or proteins, require a whole lot of energy input, mostly provided by nucleoside triphosphates, sometimes as pyrophosphates and sometimes as individual phosphate groups. Any questions for this? Okay, so we did good on time, and I think we have time to go over our paper that we were going to go over from last time. So I'm gonna stop this portion of the recording maybe. Oh, no.